So I'd like to welcome you to the CooperSurgical Fertility and Genomic Solutions Journal Club series. My name's Stephen Fleming, and I'm Director of Embryology at CooperSurgical. I shall be the moderator for today's inaugural Journal Club. CooperSurgical remains committed to training, education, and knowledge sharing. Over the coming weeks and months, we're offering an online education and scientific program to help you learn new skills, deepen your knowledge, and engage with our experts and your peers. Our Journal Club is a forum designed to promote the following activities: familiarization with the latest advances in assisted reproduction technology, encouragement of active critical evaluation of the published literature, and improved understanding of the quality and broader relevance of the data presented. Our inaugural Journal Club presenter, Dr. van Montfoort, is based at the Center of Reproductive Medicine of the Maastricht University Medical Center, where she's a senior research fellow. She obtained her master's degree in biological health sciences in 2001. During her PhD, she focused on prevention of twin pregnancies by elective single embryo transfer and by identification of morphological criteria and biological markers of embryo quality. Currently, her research concerns the effects of culture media and oxygen levels on phenotypic outcomes in neonates and health effects later in development. This article, published in the first issue of Human Reproduction Open this year, is a joint study from various universities and hospitals within the Netherlands. And it is my pleasure to invite Dr. Montfoort to now present this work, or her work, should I say, to you.
Thank you very much, Stephen, for your very kind introduction. Indeed, I am going to present to you a paper that was published in the beginning of this year on the effect of oxygen during embryo culture on the IVF outcomes. I'm currently working in Maastricht in the south of the Netherlands, but the data were obtained in Groningen, where I worked for a few years as well. I want to share with you that CooperSurgical retained me just to give this presentation, and that I don't have any other relationship with CooperSurgical, nor with any other company or decision-making authority. What is known already? Already in 1996, it was discovered that the embryo consumes oxygen at a relatively constant level throughout pre-implantation development, with the exception of the blastocyst stage, where the consumption is considerably increased. In the in vivo situation, the oxygen level ranges from 1.5 to 8.7%, as was measured in several species, including human, with the level of oxygen in the uterus being lower as compared to the oviduct. This is in contrast with the atmospheric air, which contains 20% oxygen, and which is used in the IVF, which is used in the IVF laboratories for a long time. Nowadays, incubators are available that can also adapt to 5% oxygen. But still, a lot of clinics, until a few years ago, 40% of the labs throughout the world are still using the 2-gas incubator systems with the high oxygen level. A lot of researchers were interested in the effect of a high oxygen level on the intrinsic properties of the embryo. And they found that a high oxygen level could affect the gene expression, protein profiles, metabolism, the mitochondrial membrane potential, DNA methylation, and also the development of the human embryo. Embryos cultured in a high oxygen level will develop less often into a good quality embryo. You might expect then that the live birth rate will be lower as well when you compare at a higher level of oxygen, but these results are not so straightforward. In this slide, I show you the results of 2 meta-analyses. One is from Bontekoe from 2012, it is a Cochrane review, and the other one is from Nastri from 2016, and this one includes more recent papers. Both meta-analyses only include randomized controlled trials comparing the effect of 5% with 20% oxygen, and the outcome is live birth rate. When looking at all cycles conducted in the included studies, there is a clear effect of oxygen on the live birth rate, with 5% oxygen having an advantage regarding live birth rate. However, if the data are split up according to the day of embryo transfer, and when we look then first at the data of early embryo transfer, so transfer at day 2 or day 3, the result is not that clear, as you can see here within the green circles. The confidence interval includes 1, meaning that it is not clear whether the low oxygen rate is advantageous for the live birth rate. One could argue that it might be because of a low sample sizes, but in the meta-analysis of NASTRI, almost 2,000 and 1,800 cycles were included in the groups. When looking at late embryo transfer, blastocyst stage transfer, there is a significant effect and an advantage of 5% of filtering on the 5% of oxygen for the live birth rate, as you can see here and here as well. So you might think that it takes, or you need at least 5 days of culture before having an effect of oxygen. But the group of Kierkegaard from Denmark, they did a time-lapse comparison of embryos cultured under 5 and 20% oxygen. And they found that most effects on the embryo development were found before pre-compaction stage. At the blastocyst stage, there were no differences anymore in the development. And the same has been reported by the group of David Gardner, but then in mice. So this means that the pre-compaction embryo, or the cleavage stage embryo, is more vulnerable for a high oxygen level than at the blastocyst stage. But then why don't we see an effect when we compare studies that compare the transfers of these early stage embryos? Well, that is, I think, because the effect of oxygen is not an all or one principle. It's not that all embryos are affected by a high level of oxygen. If we compare a woman who has 7 embryos, for instance, and when we would culture these 7 embryos under 20% oxygen, for instance, 2 embryos will develop into a high quality embryo. While when we would have cultured these embryos in 5% oxygen, maybe 5 embryos would have developed into a high quality embryo. But the selection of the embryo for transfer is based on the quality that we see through the microscope. So in the end, the same embryo will be selected for transfer, whether we would have cultured the embryos in 20 or in 5% oxygen. And therefore, if you only compare the results of fresh embryo transfers, as has been done in the studies before, you will not find a clear difference in your pregnancy rate. But the advantage is, in effect, that in the 5% group, more embryos of good quality are remaining, and these can be cryopreserved for later use. So therefore, I think it is better in these kind of studies to use the embryo utilization rate or the cumulative pregnancy rate, including the transfers of frozen embryos as well. Therefore, the objective of the present study, the primary objective, was to investigate whether 5% oxygen as compared to 20% leads to a higher cumulative live birth rate per IVF cycle. And as a secondary outcome or objective in this study, we wanted to assess whether different oxygen concentrations during culture would affect birth rate. I didn't put my attention to this in my introduction, but this interest come forward from our research on culture effects on the offspring, which we used to conduct mainly on culture media. The study design. It was a retrospective cohort study. We included all IVF cycles with or without ICSI performed between January 2012 and December 2013. We included only 1 cycle per patient, and this was the first cycle that was conducted within that study period. But that does not necessarily mean that it is the first cycle of that patient ever conducted. The frozen cycles were included until December 2018, so a 5 to 7 year follow-up. And in this period, 85% of the cryopreserved embryos were thawed again. The study was a transition period. We had 4 incubators in the lab, and 1 of these was set at a 5% oxygen level. As an inclusion criterion, we had that at least one oocyte should be, that there should be at least one oocyte that could be inseminated or injected. And as an exclusion criteria, we excluded cycles using donor gametes, pre-implantation genetic testing, or fertility preservation. The cycles or the embryos per woman were allocated arbitrarily to one of the incubators based on an available position in an incubator. Other clinical and laboratory procedures were similar in both groups. The embryo transfer was performed at day 2. The embryo cryopreservation was performed at day 3. And in the end, we asked for a delivery report from the midwife or the gynecologist to collect the perinatal data. Statistics. To compare the continuous outcomes, we used the student t-test and linear regression. For the categorical variables, we used the chi-square test and logistic regression. The oocyte and embryo characteristics were compared in 2 different ways. One was an analysis per cycle, meaning that first per cycle you calculate, for instance, the fertilization rate in that cycle, and then you average all fertilization rates of all cycles. And we used a linear regression for that. We also did an analysis per oocyte and embryo, and therefore we did a general estimated equation binary logistic model, and we did that to correct for the interdependence between oocytes and embryos from one woman. We also corrected for possible confounders, and the embryo and pregnancy outcomes we corrected for maternal age, cycle rank order, fertilization method, and cause of subfertility. And for the perinatal outcomes, we corrected for gestational age at birth, fertilization method, number of transferred embryos, parity, cause of subfertility, and duration of subfertility. Before we could start our analysis, we had to solve one issue. As I said, we had 4 incubators in the lab, of which one was put at 5%. But that incubator was of a different brand as the other ones. This is an issue that I notice is encountered a lot in papers on comparisons of oxygen, because not all incubators are suitable for the 5% oxygen. So often a different type of incubator is used. But yeah, in order to be sure that if we would find a difference, that it would not be due to the different brand of incubator, but to the oxygen, we decided to do an analysis of the data in the year preceding this study, so in this case 2011, when all incubators were still at 20%. And as you can see, the patient characteristics from the cycles that were cultured in these 2 incubator types were similar, had similar maternal age. The fertilization method was similar, as was the number of oocytes, but also the outcome measures like fertilization rate, cleavage rate, good quality embryo rate, embryo utilization rate, they were all the same. And this made us confident that if we would find a difference in our comparison, that this would not be due to the incubator brands, but to the oxygen. This is the flow chart of our study. As I said, we included the treatments between January 2012 and December 2013. And these were 965 patients with around 1,500 IVF cycles. We excluded the cycles with PGT, fertility preservation, donor gametes, and then we ended with 871 patients with almost 1,400 IVF cycles. We selected only the first cycle of each patient within the study period, and then we ended with 195 patients cycles in the 5% group and 676 in the 20% group. We did a follow-up until December 2018 regarding the frozen embryo transfers, and we analyzed the data on embryo characteristics and pregnancy outcome and perinatal outcome. The patient characteristics of both groups, they were all the same actually. Maternal age was similar, as you can see. The duration of subfertility, the cycle order, so which the stimulated cycle order, whether it was the first, the second, the third, or even more cycle of that patient. Fertilization methods, primary versus secondary subfertility, and the cause of subfertility. Then the embryo characteristics analyzed per cycle. These were 195 and 676 cycles. The number of oocytes was similar. The number of injected and inseminated oocytes was similar, as was the fertilization rate. However, the cleavage rate and the good quality embryo rate were better or higher in the 5% oxygen group. Slightly more embryos were transferred in the 20% group. And this is because, probably because at that time there was a rule that when a woman did not have an embryo of good quality, 2 embryos be transferred. And apparently this happens more often in the 20% group. Definitely more embryos could be cryopreserved in the 5% group, leading to a higher embryo utilization rate in the 5% group. And in the end, also more thaw cycles, of course, with cryopreserved embryos. More or less the same results when we analyzed the characteristics per embryo or oocyte. We had slightly more than 1,600 oocytes in the 5% group and almost 5,500 in the 20% group. Again, the fertilization rate was similar, but cleavage rate and good quality embryo rate were higher in the 5% group, and more embryos could be cryopreserved and thus utilized. However, the higher quality and the higher utilization rate did not significantly affect the pregnancy rates, as you can see here. Ongoing pregnancy rate and live birth rate are higher in the 5% group, but the difference is not big enough to be significant. However, if we include the transfers of the frozen embryo transfers, then the difference becomes bigger, of course, leading to a 25.1% live birth rate in the 5% group and an 18.8% live birth rate in the 20% group. And after adjustments, this is just significant. These are the results of the perinatal outcomes. We separated them for the perinatal outcomes of the singletons born after fresh embryo transfer and after frozen embryo transfer. But in both groups, none of the perinatal data that we analyzed were significant. So birth weight was similar. The Z-score is the birth weight corrected for gender and gestational age compared to a Dutch reference population, was also similar. Gestational age and gender, I said, were similar, and the same for the singletons born after the transfer of a cryopreserved embryo. So in conclusion, the culture for 2 days under 5% oxygen as compared to 20% leads to more good quality embryos, more embryos suitable for cryopreservation, and a higher cumulative ongoing pregnancy and live birth rate, while the perinatal outcomes were similar. The strengths and limitations of our study. Well, a strength was that we reported on cumulative live birth rate instead of only using the fresh embryo transfers. We had a long follow-up time of 5 to 7 years for frozen embryos, in which 85% of the embryos were thawed. We did an analysis per cycle and per embryo for the embryo characteristics. Both groups were from the same time period, meaning that all the other clinical and laboratory parameters were all the same, and we analyzed perinatal outcome. As a limitation, well, of course, I said the study was unfortunately retrospective. We did not do a randomized controlled trial. We used 2 different types of incubators, although based on the results from the year preceding this study, which did not show a difference between the outcomes of the incubators. We feel confident that the difference that we find now is due to the oxygen and not to the incubator. Cryopreservation criteria are center-specific, of course, and therewith the results might not be transferable to every clinic. Although the criteria that we used, cell number and fragmentation and the absence of multinucleated blastomeres, is used a lot in several clinics. And our study was absolutely not powered for the birth weight analysis. So for the wider implication, I would say that the results of our study suggest that the use of 5% oxygen instead of 20% is to be preferred based on the improved embryo utilization rate that leads to a higher cumulative live birth rate. I want to thank you very much for your attention, and I'm happy to answer any questions or to discuss this paper with you. Thank you very much.
Thank you, Dr. Montfoort, for that excellent presentation of your work. It's certainly generated quite a few questions, and there are some quite interesting ones for you. The first one concerns the study design to some extent, in that it appears that all embryos were frozen on day 3, but for the first couple of years, they were thawed on the same day that they were transferred. But from September 2015 onwards, embryos were cultured overnight at 5% oxygen. So the question is, could that have positively impacted the results during that later period of time during the study?
That's a good question, and good that you noticed it indeed. Yeah, well, time goes forward, and of course you want to improve your lab. So in the meantime, of the follow-up of the transfer of the cryopreserved embryos, we got more incubators that were suitable for 5%, and we switched in the end completely to 5% culture. So indeed, we could not culture the thawed embryos in 20% anymore. So from September 2015 onwards, these embryos that were initially cultured under 20% were now cultured at 5%. But this is only a very small amount of the embryos in this study, as most of them had been thawed already before the year 2015. We did not do an analysis whether these embryos were doing better compared to the ones that were cultured at 5% in the beginning also, but it could have, but I don't think it would have influenced the outcome of our study, as you would expect that culturing the embryos under 5% would be more advantage for the embryos, and that would have reduced the difference between the 20% and the 5% group, but we don't see it, so I don't think it has influenced our study much. It's a good observation.
Yeah, and there's quite a few questions about varying concentrations of oxygen. So do you have any data or any opinion on whether culture of human embryos at 2% or 3% or 5% or 7% oxygen is the most beneficial?
Yeah, as far as I know, it had the levels that you mentioned, 2, 3, 5, 7, have not been compared in one study. I know quite recently a few studies have been published on culturing under 2% oxygen, but that does not improve compared to 5% oxygen. So, but yeah, these are only a few studies, and maybe sample sizes are not big enough, but it's not a very clear improvement when you would culture under 2% at least compared to 5%. So yeah, no, I cannot be sure, but I don't think it will do much to it.
Okay, and a very common question is it okay to pre-equilibrate the media in 20% oxygen and then culture in 5% oxygen, or should you really be pre-equilibrating in 5% oxygen?
Well, I haven't compared it in a study or whatever, but my feeling would be that you have to pre-equilibrate in 5% as well, just to make sure that everything is set in the perfect setting before you put an embryo in. Yeah. Great.
And a very interesting question that someone asked is that does your data suggest in fact that if you culture at 20% oxygen, then that is more selective for greater embryo viability? In other words, it selects those embryos that are not so comfortable at 20% oxygen. And could that be a way of actually achieving the very best embryos in culture?
Yeah, that is a good question, actually. I've been thinking about that as well. Especially when I was thinking about the difference in outcome that we saw in the meta-analysis when you split up the studies according to cleavage stage or blastocyst stage. And I would have expected the opposite, that you would see a difference in cleavage stage embryos, because then the selection has not been taken place already, and that you would not see a difference anymore at the blastocyst stage, because indeed of the selection, you would expect that the embryos that suffer the most from the 20% oxygen do not survive until the blastocyst stage, so that in the end you would select the best embryo. But apparently that is not completely true. The embryos are intrinsically apparently changed, as still, and when you transfer embryos into blastocyst stage, you still see differences in live birth rate when you compare fresh embryo transfers only. So yes, to a certain extent, I think you will make a selection, but apparently, yeah, it's not completely solves the problem.
No, I can understand that. Another question is whether you looked at the miscarriage rates between the two groups of patients.
Mm-hmm. Well, then I have to go back to the slide with the, if you don't mind, with the pregnancy data, because I included as well the biochemical pregnancy rate, which is over here, which is also already higher in the 5% group compared to the 20% group. Yeah, the live birth rate is slightly higher in the difference, I would say, in live birth rate is slightly higher compared to the biochemical pregnancy rate. That would assume maybe a slightly higher miscarriage rate, but I don't think it's significant. I think it will be more or less the same. So no difference in miscarriage rate.
Okay. And another question relating to the data is, did all the patients have at least one embryo transfer?
That's a good question. Yes, yeah, I think they have. Yeah, of course, it's already some time ago when I analyzed the data, but yeah, to the best of my knowledge, everybody did have an embryo transfer. Yeah, yeah, but yeah, I cannot be sure. Maybe one or two or very few would not have an embryo suitable for transfer, but at least there was no difference between the groups now.
And I think a pretty valid question in terms of understanding the rationale for the study further is what's the specific reason for using 5% as opposed to atmospheric oxygen?
Yeah, I think it's because it mimics better the in vivo situation, although not perfectly, of course. So intuitively, you would use the 5% as that is what the embryo is exposed to in the oviduct and in the uterus. But as said, the oxygen level changes on the oxygen level where the embryo is exposed to changes on its way from the oviduct to the uterus, because in the uterus the oxygen concentration is even lower. But that would be the main reason, and also because now the data are showing that it is indeed better to culture on a 5% oxygen, especially as more embryos develop. The embryos will develop better. So for the patient in the end, it will be more advantage if the embryos have been cultured on a 5% as compared to 20%.
I guess the question was probably more concerned with the mechanisms involved. For example, are embryos cultured in atmospheric oxygen subjected to greater oxidative stress than embryos cultured at 5% oxygen?
Yeah, of course, they are exposed to a greater oxidative stress. And as has been shown before, many intrinsic properties of the embryos respond to a higher oxygen level, the gene expression, the methylation profile, etc. Metabolism within the embryo changes according to the oxygen level it has been exposed to. So all this together will definitely affect the embryo development, and also probably the aneuploidy rate seen in embryos all these factors like methylation, gene expression, metabolism are also all known to affect the chromosome stability within an embryo. And if the stability is affected, then the chance for an aneuploidy is higher. So it might also lead to higher aneuploidy rates. Yeah.
And there have been some slightly left field questions as well, which are always interesting. Does the purity of the oxygen make a difference? And will the oxygen concentration vary from countries with different weather conditions? I would have thought more different altitudes, but you know, how should people be fine-tuning their use of low oxygen?
Nice, nasty questions. Yeah, the environment. Yeah, that's an especially if you're living at a high altitude. I'm wondering whether your oxygen level in vivo is then different. I mean, the atmospheric oxygen, of course, is different, but whether the body is capable of maintaining the same oxygen, I guess it is, or at least to a certain extent. So, and I presume that the incubators, the sensors within the incubators are capable of keeping the right gas mixture within the incubator to keep the oxygen levels at the right level, even though you're at a high altitude or a low altitude, maybe. But yeah, who knows? Would be interesting to investigate, of course. Yeah. And you had another.
I think hemoglobin carries more oxygen at high altitude.
Yeah.
So I think we adapt.
Yeah, exactly. Yeah.
Increased atmospheric pressure.
Yeah.
Another interesting question, which I don't think was addressed in your materials and methods, was would culturing the embryos in a single drop or culturing them in group culture have affected the results? So did you control for that in some way between the two groups?
No, we didn't. All embryos were cultured as a single embryo within a drop.
Oh, so you did control for that?
Yeah. Yeah, we didn't have to control for it because they were all cultured in the same way. Yeah.
And there are various questions about the culture media. I guess an interest in whether the culture media itself may have had an impact, but I assume that both groups were cultured in the same culture media. And also, I'm not sure that this is really relevant, but there's a suggestion in one question that different media require different percentages of oxygen. Have you come across that at all?
No, not different percentages of oxygen, different percentages of CO2. I know.
CO2 I would understand.
Yeah.
But
no,
it doesn't apply to oxygen as well, does it?
No, not as far as I know. No.
And another question relating to not so much the purity of the oxygen, but the humidification of the gases individually or as a mixed gas. Does that have any impact on the results, do you think?
Yeah, of course, I haven't investigated it, but we did not use a pre-mixed gas, how do you say, pre-mixed gas. What I understood from using pre-mixed gas is that sometimes it's difficult to keep the right gas mixture in your incubator. For instance, if you have opened the incubator, then because the incubator cannot extract CO2 or air separately, but has to extract the prepared gas mixture. But it might take more time to get the desired gas mixture within your incubator, but I have no experience with that. So yeah, it's just what I've heard.
And probably the last question, but an interesting one nevertheless. How how do you explain almost double the multiple pregnancy rate in the 20% group? And can reducing the oxygen levels actually be one way to reduce the chance of multiple pregnancy occurring?
Yeah, I think, well, in this study, the difference between the multiple pregnancy rate was caused by the fact that in the first year of the study, in 2012, we had a policy that we would transfer 2 embryos if the quality of, if there was no high quality embryo available. And because the quality is reduced in the 20% oxygen group, we did more double embryo transfers in the 20% oxygen group, which has led to a higher multiple pregnancy rate. Nowadays, at least in the Netherlands, we in most cases transfer only 1 embryo. So then the difference in multiple pregnancy rate will not be dependent on whether you culture under 5 or 20%. But if you still have a policy in your country or in your lab transferring multiple embryos, yes, then indeed, I think that culturing under 5% might lead to a reduced multiple pregnancy rate if you take the quality of the embryos into account when deciding how many embryos to transfer. Yes.
And I think we just have time for just one more question. And again, it relates to different culture media from different companies. Can we apply 5% oxygen with all media?
Yes, as far as I know, you can. I haven't heard of any medium that says that you cannot culture under 5% oxygen, especially nowadays, since most of the labs have been switched already to 5% culture, and also the culture media manufacturers will adjust their medium for the 4 to 5%. So yes, as far as I know, there is no medium that cannot be used under 5% oxygen.
Wonderful. So thank you very much, Dr. van Montfoort, for answering those questions so well. We had very many questions, over 50 questions, so I'm sorry to those of you where we were not able to answer all of them.
Nice.
We would try and address them later on, perhaps.
Yes.
Okay. So finally, a reminder to everyone that once again, there will be another journal club next week. But nevertheless, please do provide feedback on today's journal club, and rest assured that we will send each of you a certificate of attendance for your CPD points. The third week of our Fertility and Genomics Education webinar series starts next Monday, 20th of April. The theme will be reducing miscarriage through PGTA and accurate embryo assessment. You can register for our webinar and book a one-to-one chat with an expert regarding the webinar topic. And our advice is to book early, since these one-to-one sessions do fill up quickly. Sessions will be repeated to suit different time zones, so we've got you covered. Visit our website mentioned here to book. So finally, it remains to thank all of you for attending our inaugural journal club, and we hope to see you again next week. In the meantime, please keep safe and healthy.